Thank you for the introduction, and thank you for inviting me to talk about this topic. So today we'll go over considerations for the integration of biosimilars into clinical practice in oncology. So I just need to specify that this talk was put together with the help of a scientific planning committee. These are my disclosures. I'm an equal opportunity person, so I spread the love. And in terms of commercial support, this program was put together with a grant from Hoffman LaRoche. So just to quickly go through the learning objectives, after I finish my talk, we should be able to distinguish the regulatory pathways for reference biologics, that's the original biologic drugs, from their biosimilars, evaluate the efficacy of biosimilars approved by Health Canada, and differentiate that a little bit by how the EMA and the FDA does that, and also apply relevant criteria for adopting these new drugs into our practice every day. So I'll go, in terms of the outline, through a quick review of what constitutes a biosimilar drug, as these are fairly new drugs, the regulatory requirements for the approval of these medications, how we evaluate them in oncology, and any considerations for integrating them into our practice. So biologics and biosimilars. What's the difference? To be honest, when I first heard about biosimilar drugs, I thought, what's the big deal? They're generics, it'll increase the availability of these drugs on the market, it'll drive down prices, this is great. But as I'll go through my talk, you'll realize that that's not 100% the case. So a biological medicine, or biologic, is defined as any medicinal product that comes from natural sources. So things like vaccines, blood products, therapeutic proteins like monoclonal antibodies that we use mostly in oncology are considered biologics. And these are drugs that are usually given through the parenteral route because they're proteins, and if we ingest them, our digestive system will degrade them. So how are biosimilars, so a similar drug to the original biologic, different from generic molecules, from different drugs that we take in pill form, for instance? Well, a generic is a molecule, so you can pretty much reproduce that copy for copy, whereas a biosimilar drug is produced in a living organism in cell lines. So any minute difference in that cell line or the manufacturing process down the line will produce a protein product at the end, like a monoclonal antibody, that's slightly different as the end product. So it's very similar to the reference biologic or the original biologic, but it's not 100% the same. And when it comes to evaluating these drugs, generics, essentially what you need is a small clinical trial just to make sure that it has the same pharmacokinetics and pharmacodynamics as the original drug, and that the toxicity is the same. Whereas with biosimilars, you really need to go through a more rigorous process. And what that means at the end of the day is conducting a phase 3 clinical trial in patients who have cancer to compare it to the original biologic drug and make sure that they're equivalent. So this is just a breakdown. I won't go through the whole thing of the process in terms of how biologics are made. So essentially, you engineer DNA into a cell line in order to produce your protein product of choice. So in oncology, that's mostly monoclonal antibodies. And then you produce culture media on a small scale and a large scale in order to grow these cells and purify your protein at the end of the day and market it. So any small change in any point in this pathway can create a change in the protein product at the end of the day. So a biosimilar drug, as defined by Health Canada, is a biologic drug that's similar to the original biologic that is already authorized by sale, but not identical. So these were previously known as subsequent entry biologics, and they're based on, like I alluded to, an approval process that compares them directly to the original biologic drug. And these are drugs that usually come onto the market after the patent for the original drug has expired, which is now happening for a lot of the drugs that we're using in oncology, like bevacizumab, trastuzumab, rituximab. So, as I mentioned, a biosimilar drug is not an exact copy of the original monoclonal antibody. And this is usually because the manufacturer doesn't have all of the information available to them as to how the original biologic drug was produced. So that means they might use a different cell line in order to produce the antibody. They might have slightly different manufacturing processes when it comes to purifying the final protein product. And so it's because of these reasons that they're called biosimilars and they're not identical generic drugs. So how are these drugs approved? So in terms of looking at the different levels of evidence for approving these drugs for clinical use, we have to look at many different facets. So analytical data, that is the protein structure itself, very, very basic scientific aspects, moving up to in vitro or in vivo information, and then finally looking at the clinical data like phase 1 studies for pharmacokinetics and pharmacodynamics, and finally looking at phase 3 studies to show equivalence to the original biologic drug. So these are very large-scale clinical trials involving hundreds of patients. So it takes a while for these biosimilars to be approved because, as you can imagine, organizing, recruiting to a phase 3 clinical trial and having enough patients can take a few years until you have the final information. In terms of the regulatory authorities, they look at something called totality of evidence. So what does that mean? That means that there's no one aspect of the evaluation of a biosimilar that will lead to its approval. So even if you have a phase 3 clinical trial showing that it's equivalent to the original biologic, you need to make sure that the other parameters fit, so that the protein structure is more or less the same, that the non-clinical in vitro or in vivo testing that you have is pretty similar to what you have for the original biologic. So when, let's say, Health Canada is assessing a biosimilar drug for approval, what they want to know is that all of these parameters are very similar to the original biologic drug in order to approve it. So as I alluded to, there can be multiple steps in the production of a biosimilar that if you change something will lead to a different protein product. So these are kind of the different steps, and it shows you in terms of what the risk is for having a final product that is slightly different or very different. And if you have changes in any one of these steps, then that leads to a different process for reevaluating that to make sure that the final protein product that you have is as similar as it can be to the original. So as an example, if the manufacturer changes the filter that it uses for its purification process, for instance, the risk that the final protein product will be very different is pretty low. So in terms of evaluating to make sure that that change is acceptable, you need just some analytical data or process studies. But if you change the cell line that's producing the protein product, for instance, then you can imagine that that's going to lead to a much bigger change in the final protein product. And so you need to restudy the whole process over again in a more rigorous way. So it's very, very controlled, and it's very, very specific in terms of what you need to do in order to have this drug approved. In terms of the kinds of trials that are used to determine if the biosimilar drug can be used instead of a biologic, we look at equivalence trials. So what that means is that you look to see to make sure that the drug is no better and no worse than its original biologic. So in terms of an equivalence trial, what is usually done is that you set confidence intervals at certain limits that are determined by statistical analysis in terms of what's acceptable in changes. And then you compare your two drugs and you make sure that they fall within those margins that you've preset. So if the biosimilar drug falls within your upper and lower equivalence margins, then you can say it's equivalent. But if it doesn't, you can't. So how do we evaluate biosimilars in oncology? So I'll start with a question, true or false. A biosimilar that has been studied and approved in one tumor site, for instance, let's say bevacizumab in non-small cell lung cancer, may have an indication extrapolated for a second tumor site, let's say ovarian cancer, without having to go through a second phase 3 clinical trial, specifically in ovarian cancer, to prove that it's efficacious and safe. Okay, maybe just by a show of hands, who thinks it's true? Who thinks it's false? So the answer is actually true. So what that means, and this was very surprising to find out for myself, is that if you show that let's say bevacizumab is exactly equivalent to its biosimilar for lung cancer, you don't need to study it again. It's up to the regulatory body then to decide if they're going to extrapolate the indication for use to another tumor site. So this is just to compare the differences between biosimilars and the original reference biologics. So when the original drug comes out on the market, what we're looking at is study in the intended patient population. So what that means is if you're developing a new anti-cancer treatment, then you're looking at the population that has that tumor that you want to study. And these clinical trials, in terms of their design, is usually a superiority trial. You want to prove that it's better than the standard of care that you already have. So you look at clinical outcome data such as overall survival or progression-free survival or any accepted established surrogates, and you look at the safety profile of the drug as well. Another important component, especially when we're talking about monoclonal antibodies, is the concept of immunogenicity. So what this means is that when you infuse a monoclonal antibody into a patient, they have essentially a component of the immune system that's being, or a foreign protein that's being presented to their immune system. So the body might accept it as an antibody that is natural to be in your body and not do anything about it and let that antibody be and carry on its intended purpose. Or it might recognize it as something that's foreign, so it will initiate an immune response that will eventually clear it from your body. So when you're talking about these kinds of drugs, you want to make sure that when you infuse a monoclonal antibody into a patient, that their body just doesn't degrade it through the immune system cascade. And so that's something that's studied for the original biologic. And like I was alluding to, for the original drugs, if you're going to treat a certain site with it, you have to have a study for that tumor site. So if you want to use bevacizumab in non-small cell lung cancer, for instance, you have to do a study in non-small cell lung cancer. If you want to use it in cervical cancer, you have to do a separate study in cervical cancer to prove that it's effective. Now to contrast that with biosimilars, so in biosimilars, what you're looking at is a patient population in a study that you know is already sensitive to the drugs. So the original biologic was studied for, let's say, bevacizumab in ovarian cancer, and you know that there's activity there. And now what you want to do is make sure that the biosimilar is the same activity, so you study it in a patient population that's sensitive. You wouldn't study it in a tumor site, for instance, where you know there's no activity for the original biologic drug. In terms of the clinical trial, as we said, it's an equivalence design usually with pre-specified upper and lower margins. In the study endpoints, you're looking to make sure that the population is still sensitive to that drug, and you're looking at markers of response that are usually not the gold standard for phase 3 clinical trials, like overall response or sometimes pathologic complete response rates, and that's because these are endpoints that are going to happen quicker in a clinical trial. So it's something that you can evaluate more speedily, and it's something that will give you an answer more quickly. So you also want to make sure that they have the same safety profile as the reference biologic drug, similar immunogenicity as well. And as we spoke about, once you have proven efficacy in one tumor site, you may extrapolate if the regulatory body that oversees this agrees into a different tumor site without having to do another clinical trial. So it's important to note, as I spoke about, that all biologics confer a risk of immunogenicity, that there's a loss of efficacy or safety if the immune system decides to reject the monoclonal antibody that you're infusing into a patient, and this is often done through neutralizing antibodies or cytokine release, and that sometimes these antibodies can produce a generalized immune response, such as anaphylaxis. So this is something that we're trying to avoid in terms of infusion reactions, and it's something that we need to make sure that the biosimilar drug doesn't do if its reference biologic drug didn't in the first place. So Health Canada specifically requires that there be a comparative immunogenicity assessment for the reference product, just to make sure that there's no surprises in terms of reactions of the immune response, as these are slightly different monoclonal antibodies. So how do we select the most sensitive patient population? I'll give the example of breast cancer. So when you're choosing the population in which to study a biosimilar drug, you want to make sure that that population is relatively healthy. And the reason for that is because if their immune system is suppressed, either because they've had multiple lines of chemotherapy or they have a very elevated tumor burden and they're just not very well to begin with, then you won't be able to study that concept of immunogenicity. So if the patient doesn't have an immune reaction to the drug, is it because the drug just doesn't cause an immune reaction, or is it because their immune system was too weak to mount one? So this is something very important to take into consideration when you're choosing your population to compare an original biologic to the biosimilar drug. So, for instance, in breast cancer, if you take the HER2-positive population as an example, if you're studying a monoclonal antibody such as trastuzumab, you're going to want to study a population that maybe doesn't have metastatic disease, that's more homogeneous in terms of their tumor burden. So if you take an early breast cancer population, then they'll probably have disease limited to the breast or just to the lymph nodes, but nothing in any other vital organs. And so it's a much better population to study the actual response in terms of response to the drug and also response from the immune system. So Health Canada uses, in terms of appraising authorization for other indications, the term authorization of indications. So what that means, as I explained, is that once a biosimilar has been proven in one tumor site to be similar to the original biologic, it's authorized for other indications, whereas the European Medicines Agency and the FDA use the term extrapolation of indications. So they use data from one study and one indication to justify the use of it in another indication. And the way they do that is really by a case-by-case approach. So what they do is they consider the mechanism of action of the receptor target, the interaction between the two of them. Again, they look at the totality of evidence, as I've alluded to, and they make sure that there's still an acceptable safety profile without increased immunogenicity. And also what comes into play is the clinical experience with the original biologic drug. So additional considerations for authorization, you always want to be a bit critical when you're looking at a new medicine, especially if there's a gold standard reference drug that exists. So you want to make sure that the patient population that was studied was the correct patient population. You want to make sure that the endpoints that they've chosen for their study are valid endpoints. You want to make sure that the dosage and the route of administration are something that are feasible for the patient and for the physician. And you also want to look to see if the drug was studied with any other concomitant therapies that might make it a bit different if all of a sudden you want to extrapolate it to another tumor site. You want to make sure that the study results are robust enough, and if there's any differences between the original biologic and the biosimilar, that in that study it's explicitly stated why that was and that they account for that. Another important consideration is because these phase 3 trials tend to be shorter trials just to show equivalence between the biologic and the biosimilar, that the duration of the study was actually long enough. So you might be able to see a response rate, let's say within a few months, but does it have an outcome eventually on things like progression-free survival or overall survival? And that's something that's very important to consider when we're looking at the studies that lead to the eventual approval of these medications. And then what rationale is given for the authorization of indication between populations. So for instance, a cancer patient versus a patient with autoimmune disease where these biologic drugs are used quite frequently as well. So now we'll go through an example of an actual biologic and its biosimilar. And the example that I'll give is bevacizumab in non-small cell lung cancer, which is, to me, a little bit of a stretch because this is a drug that we, in Canada at least, we don't use in non-small cell lung cancer very much. But for whatever reason, the company that was developing the biosimilar thought that this would be the appropriate population to study. So this is our sensitive population for bevacizumab. So I'll go over quickly the study that led to the approval of the biosimilar ABP 215 versus the original bevacizumab. This was a randomized phase 3 trial, was double-blind and controlled. And what it looked at was patients with advanced non-squamous non-small cell lung cancer who were on their first line of chemotherapy with carboplatin and paclitaxel and were expected to get at least 4 cycles of chemotherapy and had a good performance status. They were randomized 1-to-1 to the biosimilar drug ABP 215 versus the original bevacizumab at this dosing frequency for 6 cycles. And the primary endpoint, as we discussed, because they're just looking to see if there's a marker for response that's quick, so they can get their product approved quickly, was overall response rate. They set their equivalence margins to 0.6 to 1.5 for the overall response rate risk ratio, and secondary endpoints were overall response rate, the risk difference, in overall response rate and progression-free survival. So when you look at the numbers, you see that they're almost identical for both overall response rate and progression-free survival. And it met their criteria for equivalence. So the drug was approved based on this. And you also want to look at toxicity, as I spoke about, to make sure that changing the protein structure a little bit for the biosimilar doesn't lead to a huge difference in toxicity profile. So they looked at treatment-associated adverse events, and again, the numbers speak for themselves. They're pretty similar between the two. So when you look at what Health Canada has approved the biosimilar ABP 215 for, or otherwise known as MVASI. It's for metastatic colorectal cancer, which we do use bevacizumab a lot for, and locally advanced non-small cell lung cancer, or metastatic non-small cell lung cancer. But for recurrent platinum-resistant or sensitive ovarian cancer, you see that this indication has not been approved yet. In terms of the FDA, their extrapolation is a little bit more wide than we have in Canada. So again, colorectal cancer, glioblastoma, renal cell carcinoma, and metastatic recurrent or persistent cervical cancer. But again, it's not approved for recurrent platinum-sensitive or resistant ovarian cancer. The EMA tends to be the most permissive out of the 3 regulatory bodies, and so here you can see that it's been approved for multiple indications. So again, colorectal cancer, breast cancer, renal cell, cervical, first-line ovarian cancer, as well as recurrent platinum-sensitive or resistant ovarian cancer. So how do we integrate these drugs into our process, our practice? They're coming. I have to admit, I didn't realize before what the mechanism of using these drugs was in our everyday clinical practice. How does the pharmacy order them? Do we know what our pharmacy is ordering them? Does it make a difference? Do they just choose which drug is the cheapest? So I asked our pharmacist at my center how we go about using these medications. And she said, essentially right now, we're still using all of the originals. And MVASI just recently came out onto the market in Canada and in Quebec, where I practice, and has been approved by our pharmaceutical regulatory body. Rituximab is another one that's coming soon for the treatment of non-Hodgkin lymphomas. In terms of how we go about using these medications, what she explained to me is eventually all pharmacies and hospitals in Quebec will receive a list of medications and their pricings, and they'll choose the cheapest medication. So as physicians, sometimes we don't even have a say in terms of what our patient actually receives. So this is important because there's a notion of switching patients and interchangeability between the biologic and the biosimilar. So Health Canada's authorization of a biosimilar, they stipulate, is not a declaration of equivalence to the original drug. They acknowledge the fact that it is extremely similar and probably has the same long-term outcomes, but they don't flat out say that you can use one or the other and it doesn't make a difference. So the notion of interchangeability is the ability to change a patient from one drug to the other by pharmacists without needing the intervention of a prescribing physician. In Canada, the authority to declare two drugs interchangeable is basically governed by province, as healthcare is governed on a provincial level. So every province and territory will have its own regulations with respect to this. Switching refers to changing from one reference biologic drug to a biosimilar. So again, Health Canada has issued a recommendation in terms of switching. So what they say is that if you're going to switch a patient from the original biologic drug to the new biosimilar, that you should let your patient know and discuss this with them, and discuss with them what the implications are and what it means in terms of the efficacy and toxicity of the drug. So in terms of switching from a biologic, that's the original drug, to a biosimilar, okay, we don't really have a lot of information about this, unfortunately. So in cancer, there are no big trials of monoclonal antibodies. We do have some information from other types of biosimilar drugs, like GCSF, for instance. And what this graph is showing on the bottom are 4 arms where the patients were either left on the biosimilar, they used the original biologic, or they were switched at any interval during their chemotherapy, and that the rate of febrile neutropenia or any kind of adverse event was not much difference if you switched or left them on the drug. In terms of labeling, I'll go through this quickly, but what we know is that in terms of what goes on the actual box or bottle that the drug comes in is up to the regulatory bodies that approve it. In Canada, in terms of the monograph, it has to be explicitly stated what the properties of the biosimilar drug are, whereas in the US and in Europe, they can use the original monograph for the original biologic drug. So sometimes if there are slight differences in certain properties, it won't be accounted for in that. In terms of the names, this is another important thing, and that's because the names in terms of the generic share a common name, but the brand name is different. So being able to differentiate between the biologic and the biosimilar in terms of reporting adverse events, for instance, is very important. And also knowing which drug it is that you're giving to the patient. So the different regulatory bodies have stipulated different points in terms of making sure that the name of the product that you're using is very clear and specific to whether it's the biologic that's original or to the biosimilar. Health Canada also requires that if you're going to market your biosimilar drug, that there's a pharmacovigilance program that goes along with it. So any kind of adverse event that happens post-marketing, you need to report it. And so healthcare professionals play a very important role for ensuring patient safety in this respect. So you need to know what the drug that you're giving is. So I'll quickly go through the conclusions. Takeaway messages from this talk is that biosimilars are similar to their reference biologic in efficacy and safety, but they're not identical. Equivalence trials are sufficient for approving these drugs. Biosimilars do not have to go through a rigorous clinical trial for every tumor site in which we use them, as their reference biologic did. Switching between the original drug and a biosimilar seems safe, but we don't have a lot of evidence about this. So healthcare professionals should discuss the implementation of these drugs with their patients, and they also have a role in monitoring post-marketing for any adverse events or any changes that might be different from what they think the original biologic drug had in terms of properties. So that's pretty much it, and I'll take any questions.
Thank you. We have about 6 minutes for questions. So in terms of what you've talked about in the scope of testing of biosimilar, which is quite different from what we're used to with original drugs, you mentioned that there was a system for post-marketing surveillance, basically, in Health Canada, which might be similar to the FDA's. Does that also encompass efficacy or only toxicity and adverse events?
So it's for toxicity and adverse events, but I would urge that if anybody had the notion that the drug that they're using is not as effective as the original, that be reported as well.
And are there any situations that you know about, or is there a suggestion for a situation in which The biosimilar may not be interchangeable with the original drug for untested indications, like the ones that you mentioned for Avastin?
Yeah, so as far as we know, no, because there are no clinical trials. So unfortunately, we have to wait until it comes onto the market and we start using these drugs and we actually see. I mean, in theory, I think I made it sound a little bit more ominous than it actually is. It's just to raise a bit of a critical spirit when we're looking at these drugs. In theory, they should be exactly the same, but there are certain properties, and I didn't go through it because for Avastin, it's not such a big deal, but for Herceptin, for instance, there's more than just the target of the drug. There's notions of antibody-dependent cytotoxicity, and that might change a little bit between the biosimilar drug and the original biologic. So I think it's definitely something that we need to be aware of, and we need to know more so what we're using, because the pharmacist will issue the drug from our pharmacies in the oncology clinic based on the generic name. But I think it behooves us to know which drug it actually is that we're using in our practice.